Now I'm gonna invite Mr. Ahsan Raza from Comsets University, and he's going to be presenting on artificial intelligence in the genomes of beneficial microbes. Thank you.
First of all, I would like to thank the organizers of the conference for giving me a chance to come forth before you guys, the research community speakers and participants, to share my research and ideas. My name is Ahsan Raza, and I am a bioinformatics scholar from ComSearch University, Islamabad. My research expertise fall into three major domains, including machine learning, systems biology, and functional genomics. Talking about my research experience, I have the privilege to be part of International Genetically Engineered Machine Foundation, Boston, USA, as well as the National Institute of Genomics and Advanced Biotechnology, NICAB, Islamabad. Well, that's a brief introduction of who I am. Moving next. So, the major reason I am in front of you guys is to talk about artificial intelligence, and that is what my topic is for today. So I have divided my allocated time into two major parts. In the first part, I would give an overview of my current AI research in the domain of Parkinson disorder and its drug development. And then I will proceed towards the important part, how artificial intelligence is or can be used in the domain of genomics of beneficial microbes, the topic of today's conference. Moving next. So, starting with the current research that I am conducting here in ComSearch University, Islamabad, I will briefly cover this topic. Parkinson disease and its pathogenesis is incomplete, and that gives us the motivation to integrate omics dataset, or more precisely, the genomics and proteomics dataset of the disorder, into a network or disease network form. Then we apply different network algorithms to screen probable drug target and build a deep learning based. Note it, deep learning is a more specialized type of machine learning, and AI where algorithm learn the way human mind and its neuron learn from the experiences. So we're using deep learning to build a predictive algorithm that learns from physiochemical properties comprising of millions of values, and once it's learned, it predict the drug hit molecules of Parkinson disease. It take almost 6 months for us to complete this research, which is right now at its ending. And I believe this research will provide the research community to have more strengthened about LAG2 hypothesis, as well as the ability to fastly and accurately predict its drug hit molecules for downstream drug target development. So that's about a brief overview of my current research in ComSearch University Islamabad. The reason I share is to give a glimpse of AI work that we have been conducting here in ComSearch University Islamabad. Moving next. As the topic of today's talk is about the genomes of beneficial microbes, I will cover this new subdomain of AI in research of beneficial microbes and how it has been used. But before I jump into it, I would like to share a brief overview of the current literature review of this domain, AI and its usage in probiotics, or you can say genomes of beneficial microbes. As you can see from the graph that present the research studies from the last 3 years that contain AI, machine learning, and, well, the word "and" is very important because we're doing the literature review. So A and the and other words including probiotics and beneficial microbes are used to screen the research paper, having these words in their abstract. As a result of that, as you can see from the graph, it is evident that a total of 18 studies with 12 research papers, 3 reviews, and 3 preprints are shown. What we can conclude from this is that this AI assisted probiotic domain is in its infancy. It's starting to grow and will grow in the future, but a very good chance to use this technology and give a push start toward probiotic research. Moving next. Now comes the very important part, the most important part of today's talk, the applications of artificial intelligence in genomes of beneficial microbes. Here I shortlisted five application, or you can say usage ideas. The top three that you're seeing in the white are extracted and backed by the already research done, while the last two highlighted in the red are novel new research ideas in the domain of AI in the genomes of beneficial microbe. Starting with microbiome to disease prediction, we know for sure that there are large set of disorders in human, especially having correlation with microbiome or the gut microbiota. So sequencing the genomic content of the disease group compared to the normal group, we can train a machine learning model to accurately learn to predict about diagnosis of the disorder based on the genomic sequence of the human microbiota. That's one application of AI, and it's currently in work around the research community. Moving to the second, that is identification of probiotics properties, or you can say identification of probiotic properties of the microbe based on its genome sequence. What we can do in that domain is train a machine learning model based on different genome sequences and their respective probiotic potential as a class. As a result of that, we can accurately predict the probiotic property of a microbe directly from the genome of that organism and accurately predicting the dynamics of that property more specifically. That's a way to use AI in this domain. You can say another way to do that. Thirdly, optimized probiotic therapeutics, a very interesting application where we can learn from genomic sequence that which microbes are effective therapeutically in a certain condition and then predict the set of microbes to be used as a therapeutic against a changing condition. So changing condition is an important because that's where the prediction of the algorithm would be toward. So, in the changing condition, it do predict based on the sequence similarity of the genome of the already existing probiotics. So, moving to the fourth idea that's novel in its nature, and I'm planning to work over it, where I want to learn or train a machine learning model by giving them the probiotic property, producing genomic regions, and then after training, I would love to screen the you can say other genomic regions to see if they have the potential to be the region producing the probiotic effect. So we can screen new genomic regions having or producing the probiotic effect. Coming to the last, genome subtyping of probiotic potential region, which I personally think has the potential, is where, and then there is a need to do this, is where we can build and train a machine learning model by giving them the already annotated genomics regions of the bacteria where we think has the potential to produce the effect, and then based on that learning make new classification and hierarchical integration of the system with different type of subtype of regions that produce the beneficial effect. Now the important thing and the important part to remember here is that these subtyping are performed based on the genomic regions that we think are actually creating the effect or the emergent effect that we call as the benefit or we call the probiotic effect. In the end, of course, the results produced for these AI-based experimentations will give us the accuracy that we cannot get from statistics or traditional modeling techniques, as well as results these models will generate will be a good point of new hypothesis making that can be subjected to you can say wet lab experimentations or further validations and annotations. With wet lab, of course, people like us also need collaborations to conduct these kind of work to make them more to strengthen these researches and to prove the hypothesis that we're building. So in the end, leaving my talk here with some important points that I personally think while working in this domain, I would like to share these points is that artificial intelligence has started to provide assistance to this domain of work, and this current time is very important to start understanding its effectiveness and usage in our respective researches under this probiotic umbrella. Second important thing I think is that this technology can provide a good ability to produce fast and probable hypotheses that can be tested for further validation and increase the more knowledge bulk around the current understanding of the probiotics. And third but not last, it has the capacity to be commercialized, or you can say commercialize the amalgam of AI and probiotic research in future healthcare, which is a very, you can say, potential thing for a future. So with that, I will move to the end of my talk. If you have any question regarding me or my work and ideas, you can ask in this break, uh, or you can contact me if you want. The contact details are given here. Thank you so much.
Thank you very much, Ehsan. That was such an interesting talk.